Okay. Last time we start talking about the prokaryotes, and there is quite a few things which we need to remember. First of all is that the prokaryotes are a very simple type of the cells. For example, they don't have nucleus and other membrane-bound organelles. This is one of the very important characteristics. The DNA will be located in the middle of the cytoplasm, not protected, and usually the DNA is shorter. It's only one circular chromosome. For example, typical bacteria will contain about 3,000 to 5,000 genes. Humans will have about 25,000 genes, a little bit less than that, but so keep that in mind. We talk about the general characteristics that all of the cells will have cell membrane. In addition, they will have a cell wall. We discussed the structure of the cell wall, what makes the bacteria, some of the bacteria gram positive, what makes them gram negative. And also, we talk about some of the basic organelles. They do have organelles such as ribosomes, such as flagellum or flagella. And then we discuss a little bit about the ways how the sexual reproduction happens in the bacteria. Asexual reproduction, binary fission, they split into two identical clones, and sexual reproduction when the genetic, whenever the genetic material in the new generation is different. And we stop kind of on the conjugation. We talk about three very interesting things. The transformation, transaction, conjugation. Then we'll discuss that there is spores. Some of the bacteria will be forming spores, and those spores can withstand harsh environments, stressful environments for the bacteria. The core will protect the DNA, so to speak. And then we are started kind of, well, we haven't started on phylogeny. So phylogeny, again, we're looking at the tree of life. And the tree of life, again, prokaryotic organism, a universal ancestor, the cell which started the whole life on Earth. How exactly it happened, we're not really sure. Now, there is a few hypotheses regarding that, but they're not even theories. So in this particular experiment, over a period of time, mutations, etc., we are looking at the diversification. And whenever it's diversified, the diversified first split into two very large groups, still prokaryote and another prokaryote, the one which give us modern domain of bacteria with quite a few groups right there, and two more domains. That particular universal ancestor of that particular branch split into two. In a few billions of years, it will give us the domain Eukaryote. We'll start talking about that today and for the rest of pretty much for the rest of the semester. And then there is a domain Archaea. Very interesting group. Unfortunately, I don't have time to spend a lot of time on that, but domain Archaea are archaebacteria, also referred as. They are creatures which live in a very unusual, very harsh environment. Almost boiling water, hot springs, no oxygen the atmosphere where no oxygen is present, and so on, super salty environments, etc. So initially we believed that Archaea, and that's why they called the ancient one, because of they used to very harsh environments. Initially, we were thinking that the domain Archaea give us two different domains, Eukaryote and bacteria. Later on, with the genetic analysis, we were able to determine that domain Archaea and domain Eukaryote are quite close related, well, closer related than the bacteria itself. So there's still quite a few debates going on over there, but that's a general kind of idea behind it. So there is a few things which, a few groups. You don't really have to memorize any of those, but there is several major phyla, and you already seen some of the examples like Vibrio, cholera, right? So in this particular case, in the phylum Proteobacteria, we have the groups based on the Greek alphabet. Alpha, beta, gamma, delta, epsilon, and so on and so forth. So in this particular case, there is several groups of bacteria which we will talk about later on. One of them would be a Rhizobium right here. Rhizobium is a very interesting group of bacteria. They called nitrogen fixing bacteria. Nitrogen fixing, what does it mean? About 78% of the atmosphere right here is pure nitrogen, N2. Nitrogen is extremely important nutrient for the plant. Then the plant have to have a nitrogen in order to build up the amino acids, and amino acids will create the proteins. So far, so good. So in this particular case, plants don't have the ability to take nitrogen from the atmosphere, break it down as the molecule, and convert it into the amino acid. They have to have some sort of compounds of the nitrogen, such as nitrates. And this is what the Rhizobium as the bacteria can do. Take the nitrogen, convert it into the form of nitrogen which plant can use. Therefore, they're creating symbiotic relationship, mutualistic. I'll give you this, you'll give me that. So bacteria gives the readily available for plants nitrogen. What the plants gives the Rhizobium bacteria? The place to live. They live in roots, something which called roots nodule, and you will see those next, well, in a couple of weeks when we'll go over the plants. And within that particular roots nodules, the plants will supply them with the sugar, which is coming from the photosynthesis. Remember the group of photosynthesis. So basically, the bacteria has place to live and stuff to eat, and all it does is providing the plant with the readily available nitrogen. So quid pro quo, so to speak. You give me this, I'll give you that. And then there is few of them which are pathogenic, Rickettsia, the typhus, Rocky Mountain fever. Then we have the Nitrosomas also work with the nitrogen, but now ammonia, which is kind of toxic, well, toxic to most of the cells. They will be able to take it and convert it into something less toxic. Spirillum rat fever bite, Escherichia coli, we already talked about that, talk a little bit about the Salmonella. Arsenio pestis is basically the cause of the black or bubonic plague. You heard about those, but again, it's a bacteria, so it can be treated with antibiotics. We talk about the cholera already. There is a Chromatium sulfur-producing bacteria that oxidize sulfur. That particular bacteria is really interesting. It does not really use the photosynthesis in order to obtain organic compounds. It's something which is known as a chemosynthesis. They are using a different compounds in order to get that energy, in order to create the organic compounds from inorganic compounds. And then there is a few others, but again, some of them are pathogenic. So here we go. Now, next group of the bacteria, or next file, sexually transmitted diseases, spirochetes. We talk about the spirochetes, specifically those ones, treponema. You've seen it in the lab, but looking at the microphotograph, you can clearly see that it is the spirochete. Cyanobacteria, discuss those ones. This is the ones which probably started the photosynthesis, and look at this one. They are responsible for generating half of the world oxygen, meaning the oxygen which you are breathing right now most likely has been produced, at least half of it was produced by some sort of tiny little bitty bacteria. There is just billions upon billions of them. So, and then we'll talk about the gram-positive bacteria, specifically anthrax. So in this particular case, you might notice that gram-negative bacteria, many of the gram-negative bacteria, which at least we covered in our lab, are pathogenic, but not all of them. So some of the gram-positive bacteria, like anthrax, they they will create, well, causes anthrax, diarrhea during the antibiotic therapy and so on. So there is quite a few of those particular interesting bacteria. Unfortunately, don't have much time to talk about that, but please remember, shape is not the phylum. It helps with the identification of the bacteria, but you can find the round ones pretty much cocci pretty much everywhere. Same thing with the rod ones or spirochete ones and so on. So ecology. Ecology is very diverse. It's probably one of the most diverse group we know, even though we haven't discovered all of the species, but some of them are aerobic. What aerobic means? Aerobic means that they need oxygen. Why they need oxygen? Cell respiration. There is a group of the bacteria which are anaerobic. Anaerobic or strictly anaerobic bacteria, they do not like oxygen at all. As a matter of fact, oxygen is toxin for them. So they live like in our intestines. This is where not much of the oxygen goes in swampy areas, deep in the ocean, etc. And then there is a facultative anaerobes. They tolerate oxygen, they can use oxygen if the oxygen is present, and if there is no oxygen, they switch to some other form of something which is known as fermentation rather than cellular respiration. Energy, already kind of talk about that during the lab. Heterotrophs versus autotrophs. Heterotrophs have to take the organic compounds by breaking them down. They will release the energy. Cellular respiration is one of those particular ways to do it. And then there, of course, there is a fermentation. Cellular respiration is much more efficient. And then, of course, we are humans, animals are heterotrophs. Autotrophs, taking carbon dioxide and with the help of specific reaction and the source of energy, like light or any other chemical reactions, they might ended up with creating the organic compounds. So that particular organic compound of food for them, therefore autotrophs means food by themselves. They are making them. And within the autotrophs, you can see the photoautotrophs like plants. Well, plants are not bacteria, but we're talking about cyanobacteria. They're using the light in order to make the food. And then there is a group of bacteria which lives deep in the ocean, typically deep in the ocean. There is not much of the light, so they're using a special chemical reactions, so chemical autotrophs. So anyway, here we have some of the important roles. We already talked about the nitrogen fixation. We already talked about the recycling. Bacteria are great recyclers. Along with the fungi, they are the recycle of nutrients in the ecosystem on the planet. If there will be no bacteria slash fungi who are recycling the dead material, essentially, there will be a layer of dead creatures, plants, etc., for about 3, 5, 6, 10 miles deep. And of course, it will deplete the planet from the nutrients. So they are decomposers. That's one of the very important roles, being a decomposer. As a matter of fact, typically, what we will see, you will see quite often whenever we're covering a specific group, there will be a question on the lecture exam, most likely like a short answer. What is the ecological role, or what's the primary function of that particular group? Like, what is the primary function of the bacteria? Well, you can say they are decomposers, recyclers. Same thing with the fungi, but also if it's asking you about like how it's important for humans, well, there is a couple of things, and one of them is food. Whatever it is, we're trying to eat as humans, like that. And then, of course, any kind of medicine, we are using it in order to improve our life. And then, if possible, for bacteria, it's not, but we are using quite often things like plants for building materials in the industry, so to speak. So decomposition is the very important part. So here what we have, there is a several, you don't, once again, don't have to memorize all of that, but in this particular case, nitrogen, we're breathing in, breathing out. The nitrogen by itself plants cannot use, but we have a very specific nitrogen fixing bacteria, which will change it into ammonia and later on into nitrites and nitrates. And those ones can be absorbed by the plant and they will start building the amino acids, and those particular amino acids will be building plant proteins. Then the animals coming in, chewing on the plant, taking, well, digesting the plant material, plant proteins, converting the proteins into the amino acids, and then they're building their own, right? So this is how generally it works out. Let me put the scanners away. So anyway, and as you can see later on, whenever it dies, some of the nitrogen will be released. So we're looking at the cycle. Same kind of idea happens with the carbon cycle, carbon dioxide present in the atmosphere. There is a sources of, well, there is a lot of people saying global warming, etc. How we are contributing. Yeah, definitely we are contributing to the carbon dioxide, but what carbon dioxide is? It's a starting point of photosynthesis. So by burning the fossil fuels, what do? We are creating additional food for plants, literally, or for the cyanobacteria, and they will create more food and therefore they'll create more oxygen. So everything is good on this side, right? Yes. But unfortunately, humans are quite curious creatures. What they will do, not only they will burn the fossil carbon in form of coal as well as the oil, but also we're cutting down those particular trees, we're polluting the ocean, so we are reducing the amount of consumers of carbon dioxide. And that's one of the major problems right there. So it's not only that we are burning fuels, we're also killing the organism on the other side, the one which can absorb that particular carbon dioxide. Anyway, that's it for the bacteria. Like I said, will go really, really fast. And today we'll continue with the next group, which is protists. Let me close that down. Actually, go with that. Oops. 5. Right there. So prokaryote. Oh, no, that's not it. And that's not it. There we go. And in this particular case, we're looking at the protists, and protists are eukaryotic organisms. Yes, they kind of look like a bacteria, but one of the huge differences that this is the organisms which have the nucleus, and then of course are quite complex membrane-bound organelles, mitochondria. Some of them will have a chloroplasts, and so on, so on and so forth. So now this is the overall representation of something which is known as the eukaryotes. And as you can see, there is quite a few branches coming out, and some of them are rhodophyte. I will talk a little bit about those today. That's especially in the lab. Red algae, and we'll talk about the alveolites. We'll talk about radiolarias, chromista, green algae. So you see, this is where we're looking at the plants. This is how the plant came out, or kingdom of plants. So those ones are all combined into a single kingdom. Then we have a plant kingdom, kingdom of fungi, and kingdom of animalia. As you can see, the kingdom of fungi and animalia are much closer related than, let's say, fungi and plants, even though they kind of look alike, kind of. Yet, as genetically speaking, we are talking about fungi, and animals are very close related to each other. And there is another group of protists known as the Flagellates. Please remember that particular group, because koanos means color. collar and the flagella. They have a very interesting structure. I'll show you the pictures, but this is kind of showing you the cell. There is a collar, and then there is the flagellum. Anyway, so in eukaryotes, so remember, we have three major domains: bacteria, eubacteria, true bacteria, which has about 20 or so kingdoms. We're not going into that. Then there is an Archaea with 5 to 7 kingdoms. And then we have a domain Eukarya. The rest of the semester will proceed with the domain Eukarya. And this is 4 kingdoms which you need to know: Protista, fungi, plants, and animals. As a matter of fact, next lecture we'll talk about the fungus. And then next unit, unit 2, we'll talk about the plants. And then the unit 3 is all about animals. And unit 4 is pretty much animals, but from the anatomy and physiology point of view. So in this particular case, fungi, plant, and animal are very well defined. In order to be an animal, you have to do X. Well, you have to have X and X and X, very specific characteristics. In order to be a plant, there is a very specific characteristics. In order to be a fungi, here's the characteristics you should have. Then that particular organism will be in the kingdom of fungi. What about the protists? Protists are not. Protists are polyphyletic, means that basically the eclectic group. If we find the organism which is not exactly an animal but not exactly a plant either, today we'll talk about Euglena, for example, then we're like, it's not fitting. It has the characteristics of that and this, therefore we'll put it in a group of the protists. So essentially right now, protists are a eukaryote, which is not plant, animal, or fungus. All right. So keep that in mind. It's a very eclectic group, and because of that, we'll see quite a few creatures there. Some of them will be unicellular organisms, some of them will be colonial, some of them will be multicellular organisms. Here we're looking at the picture of the brown kelp, and brown kelp is one of the largest, well, plant-like protists. We're talking about 150 to 200 feet long, one of the very important ecological protist algae. And then, although many of them are very important, but that one is creating the kelp forests up on the west coast, which is support most of the life there. So as you can see, they have, since they're eclectic, everything is put there. So we're looking at a very diverse group. And we'll talk a little bit about the different groups within, but again, don't have the time to go in very neat details. Last lab, we kind of talked a little bit about the anti-symbiotic theory. And the symbiotic theory is suggesting that there is 2 organelles, at least 2 organelles, mitochondria on the left and the chloroplast, which is probably came or been engulfed by the cell, and at some point of time, long time ago, they were about 1 billion or so, maybe 1.5 billion, up to 2 billion of years ago. They were free living cells which got engulfed by the bigger cell, and instead of destroying, they kind of start living together. Symbiosis. Sym means together, bio means life, living together. So why do we think so? Well, first of all, mitochondria as well as the chloroplasts have their own DNA. They are semi-autonomic. They can divide in a process very similar to binary fission on their own without the nucleus command, so to speak. And the structure as well as the size look very similar to a bacterium. So anyway, the anti-symbiotic theory, in brief, saying that proeukaryotic cell, which is basically a large cell, DNA, which was surrounded later on by the membrane and the fold-ups of that particular membrane were created in the plasmic reticulum. You probably remember the rough versus smooth. Rough will be dotted with the ribosomes, smooth don't have much of the ribosomes. And then aerobic bacteria got engulfed, but because it was making a lot of ATP, adenosine triphosphate, by breaking sugars, that particular big cell decided not to destroy. And then that particular big cell also engulfed the photosynthetic bacteria, probably within the cyanobacteria, and again decide not to destroy. For one or another reason, now we're looking at the branch, which eventually give us the plants, but in this particular case, that one needs sun, carbon dioxide, water, creates sugar. This one takes sugars, break down, and producing a lot of ATP, which can be used within the cell. And that's a very, very simplistically talking about the anti-symbiotic theory. Once again, we have a sometimes it's referred as eukaryote, sometimes it's referred as a prokaryote, but large cell, probably not a prokaryote. I think it was pro-eukaryotic, before eukaryotic cell, so to speak, but ended up with within the cyanobacterium, and then we start looking at the cells. What's really important to understand that particular process happened most likely several times, especially in the Protist spectrum. We have some of the algae, Protists, which have a green pigments and chloroplasts. We have, well, in the Plastids, they have a chloroplast. In some of them, they have a secondary endosymbiosis, and they will end up with engulfing something else. So in this particular case, those particular plastids will help them with photosynthesis at the different depth. Today we'll talk about several groups of the algae, such as green algae. Everybody understand that, chloroplast. We'll also talk about the red algae. We'll also talk about the brown algae. Why they called red or brown? Because literally they're using a slightly different set of plastids to perform the photosynthesis. And the next picture, here we go. We're looking at the red algae and the green algae. They're probably close related to each other, and the green algae most likely is the branch which eventually give us the plants. But the red algae are similar in DNA sequences, so they're close related to the green algae as well. But in this case, why they're red? They're using a different pigments, not chloroplasts, but some other organelles to perform photosynthesis at the deeper layers within the water. So diversity in motility. Some of them do not move at all. If they're moving, there is a 3, and you need to know, and we'll talk about that in the lab as well. There is a 3 main ways how the creatures are moving. Flagellum, I'll start with the very right one. Flagellum, everybody's kind of familiar with that. We're looking at the euglena, there is a flagellum, it's beating and it's moving the organ. Everybody's happy, everybody understood that, there is not a big issue. In this particular case, there is one actually issue. Here, look at the euglena right here, right? That's the euglena. There is a top part with the flagellum, there is a bottom part, kind of the pointy one. Which side, the top or the bottom, is the front end of the euglena? Please answer it in the chat box as the top, bottom, TB, whatever you can think of, but yeah, kind of give me your ideas. Which one is the front? Is this is the front and it goes into, or that is the front and it goes this way? So flagella is on the back. That's an interesting bottom. Bottom is the front end, so flagella is on the back. Top, okay, so now we have 2 different ideas. Some of the people saying that the flagella is on the back, so the bottom would be the front end, and we have another group of people who are saying, students who are saying this is the front, the top is the front. So any other students care to We have top, okay. Bottom, so about top, bottom, bottom, top, yeah. About half and half, 50%, well, yes. Well, 50% of you right, 50% of you is not entirely right. The front end of Euglena, is where the flagellum is. Yes, Euglena is weird in this particular sense, because in this particular case, what happened that the flagella is kind of going into the environment and pulling the body of the Euglena behind it. Many of you who probably says that the flagellum is on the back, if you can imagine, there is some creatures like sperm. Sperm has the tail, the flagellum, on the back, and it's beating, and then the sperm is moving forward. But in this particular case, think about, like, let's say propellers. All right, propeller, for most of the airplane, propellers are in front and they pulling the airplane in. Sometimes there is several versions of the airplanes when the propeller is on the bottom and it pushes the airplane. But let's say the boat, the propeller on the bottom, but also will be on the back. So this is kind of, keep in mind that front end of the Euglena is where the flagellum located. So in this particular case, not much of the difficulties, well, besides that tricky question a little bit. Now, amoeba. Actually, let me kind of skip to the Paramecium because it's a little bit easier to understand what the Paramecium has. Paramecium has tons of tiny little bitty cilia. Cilia are much shorter than the flagellum. Flagellum usually are longer, about the length of the body of the creature. In this particular case, there will be a small projections, but they are beating at the same time. And because of that, Paramecium will receive a much faster punch into a moving, and that would be the front end. So the Paramecium will be much faster moving than Euglena, because that one has a very long but slower moving flagellum. That one has tons of cilia which are beating in unison. As a result, it can propel the organism. Now, amoeba would be the slowest one. It does not have cilia, it does not have flagellum. However, it has a very curious way. It creates something which is known as a pseudopods. Pseudo means false, podos means slag, right? So pseudopod. Pseudopod is literally the extension of the cytoplasm. Whenever the amoeba think to go like, something yummy is there, it literally will start putting its cytoplasm out, and it continue to move, and then it will start grow the false leg or false feet. In this particular case, yes, that's the extension of lobes of the cytoplasm. It does not, and that's one of the reasons why it's called amoeba, because it does not really have the specific shape. It's changing the shapes. Amoeboid movement, very interesting way. So let's kind of look at the Paramecium. Today in the lab we'll go over that. So as any kind of cell, it will have a cell membrane. Okay, inside of the cell membrane, it will have the cytoplasm. That's done. Now it will have a nucleus. Most of them will have a nucleus. As a matter of fact, Paramecium will have two nuclei, the large, known as a macronucleus, and the tiny, known as a micronucleus. Those ones are important. Macronucleus are typically govern all of the process within the cell except the sexual reproduction, and this is where the micronucleus take over. So to speak. So in this particular Paramecium, are moving really fast. It has something which is known as a mouth. So basically, whenever it moves, it's scooping like a bacteria and smaller algae, smaller protists inside. And then whenever it gets into that, it will be surrounded by the plasma membrane. And now we'll end up with vacuole. vacuole which contain food. That particular food, well, the food vacuoles will be infused with the digestive enzymes, and that particular food vacuole usually travels along. While it's traveling along, the food inside of that vacuole will be digested, releasing the nutrients inside of the cytoplasm to a nearby organelles. And then eventually it will merge at the end. Whenever it's non-digestible, it will merge with the cell membrane, expelling whatever is undigested. In addition to a typical, there is endoplasmic reticulum, mitochondria, so all that particular good stuff. But in addition to that, they have a very interesting structures known as a contractile vacuole. Contractile vacuole, contract means become smaller, right? So contractile vacuole is essential for most of the protists. Most of the protists will live either in aquatic or very, very wet, humid environment. So in this particular case, what will happen, the water will start moving in, seeping in through the cell membrane. And without that particular contractile vacuole, what will happen is water will increase, and eventually the cell will burst. Contractile vacuoles help the cell to maintain the concentration of the water. It will have some channels which will gather the water, and the water will be seeping in into a central part, which is actually a contractile vacuole. As a result, the contractile vacuole initially will start increasing in size as the water seeping in, increasing, and then it will burst up, releasing the excess of water, and it will collapse. So it will contract, and then the process will continue. Again, getting excess water, bursting up, expelling water over and over. Because Paramecium is very, very fast comparatively to the other protists, it has actually two contractile vacuoles, because more faster you're moving, more exchange with the environment happens, more water will be able to penetrate or move in. So yes, contractile vacuoles allow us to excrete excess water. Extremely important, and technically that probably a contractile vacuole, and that most likely a contractile vacuole. So now, ecology. Again, most of them are aquatic or near aquatic organism. Could be heterotrophs, could be autotrophs, could be parasites, multicellular, single cellular, colonies, everything. Some of them photosynthetic, some of them are heterotrophs, so phytoplankton and zooplankton. Reproduction. Now we have something which is known as the asexual reproduction, and that's a typical mitosis. Unlike bacteria, bacteria went through the binary fission, but in this case, eukaryotes have the nucleus. Protists are a group of kingdom within a eukaryotic domain. As a result, they'll go through the typical mitosis, the division of the nucleus and then division of the cell itself, right? And you remember prophase, metaphase, anaphase, telophase, the alignment of the chromosomes, and then splitting them, and then splitting into two cells, hopefully equally. And then there is a sexual reproduction. Again, in the sexual reproduction, we are looking at the next generation, a generation which comes out from this particular type of reproduction will be genetically different than the parents. So, and it's very diverse, extremely diverse. There is so many different ways how they exchanging the DNA. It's not even funny. However, we have only one lecture, so I don't really have much time to talk about it. We'll talk about a very specific sexual reproduction within the Paramecium, and you will see it, well, you kind of will see it in the lab. So anyway, now let's take a look at the classification. Again, we're looking at the whole domain of eukarya, right? Common eukaryotic ancestor, start splitting up into several major groups. This particular group, Apistoconta, this is where a fungus as well, or fungi, as well as animals belong. There is a few other groups, and Flagellates is one of those, which probably give us, well, the colony of the Flagellates eventually evolves into what we know as the panda animals. So a very, very important group. There is a few others. There is a super groups. There is either 6 or 7 of them. I think your lab is talking about 6, so we'll keep it as a 6. So there is an excavator. A very interesting group contain euglena, and we'll talk about the euglena in a lab, and well, today and in a lab as well, in a lecture and in a lab. Then there is a chromo. Chromo means what? Chromo means color. This is the colorful one. Why the colorful one? Because many of them are photosynthetic because of the different pigments. We'll look at the golden algae today in the lab. We'll talk about the brown algae. We'll talk about the diatoms. And then there is a rhizarians. Rhizarians are basically very close to amoebozoids, and we'll talk a little bit about the radiolarians today and foraminifers, or forearms. Then there is an Archaeplastida. Plastida means that they do have some sort of chloroplast-like structures, but chloro, remember, chloros means green, and we have a chlorophytes, green algae. That particular group was specifically green algae within the group within the green algae known as the charophytes most likely give us the land plants, and we'll talk about that next week after you'll take your exam. And then, of course, we have a red algae right there. And then we have slime molds and amoebas. We'll talk about the amoebas, and I'll show you some of the pictures of a slime mold I have in my backyard. Not all the time. It's periodically shows up, and I'm kind of feeding it so it stays within the place. So, yeah, just for fun of it. And it's completely harmless, even though it looks kind of weird. So anyway, let's kind of look at the first supergroup known as the excavator. Single cell organisms, very important. They use one or several flagella, one flagellum, several flagella. Many of them parasites, some of them have the chloroplast, so they like plants, but they not. And they have something which is known as an eye spot. Today in euglena, you will see that particular eye spot. It's kind of reddish spot. It's a photosynthetic. It's not really an eye, but it's a photosynthetic. Well, it helps. It's a photoreceptors. It will be able to detect the presence of life, and because they are chloroplast, well, they are photosynthetic. Some of them are photosynthetic. They prefer to have or to go where there is a lot of life. So diplomonadas, generally this is the group who are parasites, including the humans. They have 2 nuclei, ER, endoplasmic reticulum, as well as a Golgi are not really defined. What does it mean? They don't really have it. Why not? They're taking all of the lipids as well as the proteins from the host. Then we have today, I think we have a Trichomonas. Yes, we have a Trichomonas, even though not vaginalis. But oh no, we'll talk about the Trypanosoma, but it's the same group. Parabasalia, no flagella. Well, addition to the flagella is undulating membrane. You will see the undulating membrane in Trypanosoma. What does it mean? The flagella, all right. Usually it will kind of come out from the front and it will go around the body, and there will be a membrane between the flagella and the body of the protist. Why? Very important. Usually those particular creatures, the one with the undulating membrane, in a viscous environment than just the water. Trichomonas vaginalis live inside of the vagina in the mucus of the vagina. Mucus is much denser. Therefore, it needs additional help to swim around. Trypanosoma also also has undulating membrane, lives in the water than the water, therefore it needs extra membrane, extra space to move through. So in this particular case, euglenozoids are free living and photosynthetic, and we'll talk about the euglena today as well. All right, so let's see. Oh, something happened. You will see that particular picture. I need to update it. But in this particular case, we'll talk about that particular picture in the lab, but essentially what we'll see, there will be, you see that particular kind of membrane right here, that's the undulating membrane. So here we're looking at the creature's red blood cells, plasma, right? And that would be the trypanosomes. So in this case, we'll see a very large mitochondrion, very large, we'll see the nucleus. And you see here we have the flagellum, which is kind of going around the body, and there will be a membrane, so undulating membrane attached kind of to the side. And this is what essentially you will see today in the lab, those particular squigglies. That's a scientific term for you right there. But in this particular case, this is the Protist, which is inside of the blood sample. It's not a human blood, it's a red blood, but if you'll see something like that in the patient, that patient has lots of issues. I mean, that patient most likely will die. It's treatable, but in that particular amount of the trypanosomes in the sample would be kind of hard to treat. So now, chromoalveolate may be a single cell, but could be a multi-cell. So they use cilia's, could be colonial, could be parasites. So in this particular case, within the chromoalveolata, we'll have several major groups. Dinoflagellates. A very interesting group, we have it in the Gulf of Mexico. Phytoplankton reproduce relatively easily. They have the shell made out of cellulose, and they bioluminescent. If you'll ever be kind of like in June or July, and I don't know, with the COVID right now, it's kind of hard to travel abroad, but if you'll ever ended up in the Cancun area in Mexico, the dinoflagellates are kind of blooming during the end of the June, end of May, June, and probably in the beginning of the July. So periodically you will see the bioluminescent, you will see the glowing of the waves. They're really pretty. So, and whenever they reproduce too much, they'll create red dyes, and that's not good. Actually, we'll talk a little bit about that. I'll show you some of the pictures. Now, epicomplexians. This is a predominantly parasites. They do not move, and they are probably one of the, well, I think the Plasmodium is #1 human killers in the world, the surpassing bacteria. Now, we already talked about the ciliates, or a little bit about the Paramecium, and one of the characteristics, they have 2 nuclei, large and small, and small plays important role in the sexual reproduction. Now, diatoms, we'll talk about those one as well. The shell, and you will see di means 2, so the shell is made out of 2 parts, and they're very pretty. They kind of glassy looking one, because the shell will be made out of silica, sometimes referred to as silicon dioxide. Anybody knows what the silicon dioxide is? Where you can find, if I'll say, hey, let's get some sample of silicon dioxide. Where can we get the silicon dioxide? Anybody? Any ideas what silicon dioxide is actually? SiO2? No ideas. Nope. All right. Even though all of you seen it, I'm telling you, all of you seen silicon dioxide. Silicon dioxide is the most common compound on the face of the Earth, on this particular planet. And basically, if you look through the window, you look at the glass, and my glass is not very good. clean so I can see the dirt on the glass. Glass is made out of silicon dioxide. Most of the sand, specifically silicon sand, not the coral sand, but the sand, typical sand is made out of silicon dioxide. And then there is a golden algae. Main pigment is not chlorophyll, not the green one, but fucoxanthin. So it's masking the chlorophyll. They do have a chlorophyll as the chloroplasts, but fucoxanthin masking it. As a result, we'll see a slightly different color. It will be a kind of goldish color. Then there is a brown algae. Some of them are much longer than, some of them are multicellular, and you see 50 meters, it's about 150 or so feet. Fucoxanthin also masking the chlorophyll, and that's considered to be one of the most complex. Sometimes it's referred as the algae. There is a algology, which is science talking about the algae. And then there is all mycotes, which is molds as well as the mildews, and we'll talk a little bit about that because they were very important at some, well, they are still very important, but they are really, they used to, well, I'll get to that. Historically, it was very interesting how tiny little bitty protists force humans to do different things. So sexual reproduction, let's kind of, don't have to memorize it, just know that in the Paramecium, we have something which is known as a macronucleus, micro, tiny, and that one which is responsible for sexual reproduction. Two parameciums will get together and they will create something which is known as a cytoplasmic breach. They literally merge the cytoplasm. During that particular time, we'll see that each of the parameciums will start dividing the small nuclei. As a matter of fact, it will go through the process of meiosis. And remember, meiosis, two divisions, nuclear divisions, and it will produce 4 nuclei. So in this particular case, this is why we have 4 nuclei right there. So in this particular case, 4 nuclei, and 3 of them will disintegrate, break down. Then we'll have the micronucleus will divide by mitosis. So we have the genetic variants has been produced by the meiosis. Now those particular micronuclei, some of them will disintegrate, some of them will continue. Now that one will create exact copy of itself, but that particular micronuclei is different, genetically speaking, than the previous ones because it went through the process of meiosis. And then they will exchange the micronuclei. Now, later on, micronuclei will fuse together. So haploid and haploid will create a diploid micronuclei. And then those micronuclei, the new ones, will go through the 3 rounds of mitosis, creating 2, 4, 8 micronuclei, okay? During that particular time, the macronucleus will disintegrate, break apart, and the 4 micronuclei will merge together, creating a macronuclei. All right, and then we'll end up with the 2 rounds of the cell division, will produce 4 cells total from each one of those. So as you can see, quite complex and start looking very similar. We already have the meiosis, we have mitosis, all that particular mix. So very, very important to understand that during the sexual reproduction, the genetic makeup of the parent is different than the genetic makeup of the daughter cell. So now let's take a look at the dinoflagellates. This is how it looks like. The shell is made out of cellulose. Typical, once again, typical phytoplankton. They being fed, well, they being a trophic level to be fed by the zooplankton, and then the zooplankton will be eaten by the smaller fish, and so on and so forth. Here we're looking at the bioluminescent. You can see the waves are nice and glowy, and occasionally, it might start overgrowing blooming, so to speak, and they will turn into literally red tides. Why? There is so many of those particular creatures there that the water changes its color. That's actually the picture from the coastline of Florida. Couple of years ago, latest red tides, if I remember correctly, in the Galveston we had in the summer of 2013. It's very, very dangerous. First of all, those creatures are slightly toxic. Whenever we have tons of them and the fish gets them, they become, the toxins accumulates, and eventually if you're eating, you can get sick. Also, what happens whenever those creatures are mass reproducing, super reproducing, they depleting the oxygen from the water. Typically, it happens in a warm warm time of the year, so the water is already low on oxygen, and those creatures start reproducing a lot and go through the cellular respiration using tons of oxygen. As a result, the larger creatures, fish, any kind of crustaceans, etc., start dying out because they're literally suffocating to death, and then of course they'll end up on the shores. So yeah, not a very pretty picture. Anyway, next group known as the apicomplexians, and this is the creature. And apicomplexians, according to CDC in 2015, about 200 or so millions of cases of malaria occurred worldwide. And again, many of the scientists saying, well, 200 million of reported cases. Many of them saying about 2 or 3 times, up to 5 times unreported cases, meaning up to 1 billion of people had that particular parasite, that particular disease. And in this particular case, it's treatable. We know how to treat it if we get it on time. But about 500,000, and again, if we're multiplying by 3 to 5, over 1 million, up to 2 million of people die from that particular parasite, even though, once again, we know more or less how to treat it. We know the symptoms. We know that there is the fairly effective drugs. However, those particular people lives in something which known as economically disadvantaged areas of the world, such as Southeast Asia, some of the African countries, and some of the Central as well as Latin or South America. And therefore, some people don't have the access to the drugs, or it's too expensive. All right, so yeah, that's a sad part of that. But in this particular case, we have a mosquito which bites. Whenever it bites, it's drinking the blood, and whenever it bites, it's also injecting the prey, the human, with its saliva. That's why later on, if a mosquito beat you, you're scratching. That's the reaction, local allergic reaction to the proteins in the saliva of the mosquito. Not all of the mosquitoes carries that Plasmodium. So anyway, in this particular case, as it's injecting the saliva, it's also injecting those parasites which goes into the bloodstream, and then they start reproducing within the blood, specifically infecting the red blood cells. And we'll talk a little bit about that later on. Now here we have some of the diatoms. As you can see, they are made out of glass, literally silica dioxide, and this is a picture of the diatoms you will see in the lab. This is the brown algae, kelp, and as you can see, the reproduction cycle is very similar to what we'll see later on in plants. And now those guys, Oomycotes, the water mold, so to speak. Well, everybody can understand that this is a potato which is infected with those Oomycotes, of those particular protists. In 1800s, like mid of 1800s, you know that there was a huge influx of the Irish population coming here in the United States. Why? Because all over the Europe, essentially, there was pretty much a very, very bad environment. Well, it was a good environment for those guys to reproduce, especially in Ireland. But it wasn't only in Ireland. There was a famine going on throughout the Europe. We're talking about the France and the Germany and Poland and Russia. But specifically in Ireland, predominantly lots of people were depending on the crop of the potatoes. They were growing it over the summer essentially, then they were getting them and eating during the winter. So everything is fine. However, those particular years, there was like 1845, 1846, somewhere in that area, we had a fairly cold summer with a lot of rain. Fairly cold summer, a lot of rain. As a result, those protists start reproducing and they start infecting the potatoes. So people, well, those potatoes were literally rotten in the ground, but being eaten by those mycots. As a result, many of the, literally millions of people starved to death, and some millions of people had to move, including United States. And because of the mycots, a completely brainless, tiny little bitty creature forced millions of people to move to somewhere else, including the United States. This is how United States that's one of the reasons why United States got a fairly nice Irish population. And of course, we are celebrating the St. Patrick's Day and so on, just because of that thing right here. Anyway, Rhizaria. There is two large groups. Radiolarians made glassy internal skeleton. And then there is a foraminifera. Glassy means silica dioxide. The foraminifera also forms the sedimentary rocks, but the shells is made out of calcium carbonate. Okay, two different forms. So now you can see here we're looking at the radiolarians. This is the inside. The body kind of goes around that, and those particular holes has the legs or sticking out, and they are capturing whatever they can capture. And here we're looking at the calcium carbonate shells. As you can see, they're quite large. Some of them can be seen by naked eye. One of the largest protists as well. Archaea plastida, we'll talk about the red algae. We'll see them today in the lab. Later on, you would need to be able to recognize the red algae. Don't have to say which one, but say, oh, that's a red algae. And in this particular case, they are red because, once again, they have that particular pigment right there, phycobilins. And those ones are important for pectins. Essentially, we are consuming some of the red algae as well as the brown algae. Chlorophyte, we eating those as well, we as humans. Single cell, colonial, as well as multicellular. They are really important from the point of view that they most likely give us the branch to modern land plants. And this is what we are talking about during the unit 2. Those particular three groups descended from the same ancestor, just took different routes, so to speak. Anyway, this is the picture you will see today. This is something which is known as a Colonial Volvox. That's a green algae, as you can see, literally green. And you will see the mature colony, also known as a parent colony. And there will be a smaller colony known as the daughter colonies inside. And here we're looking at the red versus green algae. You can clearly see why they call the group called the red algae. Now, amoebozoids, amoeba-like creatures, are using the pseudopods. Amoeba proteus is one of the common protists you can find pretty much in a drop of pond water. Anti-amoeba histolica is the parasite, which also can be found in the water. And if you, by any chance, drink that particular amoeba, the cysts of amoeba, it goes into the digestive system and create a huge problem for your digestive system. We're talking about the dysentery. Anti-amoeba histolica create a dysentery, is basically throwing up and diarrhea, severe diarrhea. So very, very very dangerous. Left untreated, the person will die from the dehydration. So anyway, so if the water source is questionable, it's anywhere in the world. If you're not really sure, boil it. Because I had in my army time, we had the unit next to us were drinking from we were deployed in the middle of nowhere, and unit next to us, they were drinking from local spring. Several of them ended up with that particular thing, ended up in a hospital, and in the hospital each one of them lost about 20 to 30 pounds. So yeah, because you cannot eat, nothing is holding on in your digestive system, so you're throwing up or diarrhea right away. So anyway, cellular slime molds as well as a group, because they look like mold, hence the name. However, they can create a coordinated mass known as a plasmodium, and they will start moving around slowly in this particular case. And once again, very interesting. Then they split apart. So a very, very curious group of organisms. And in this particular case, the plasmodium will create a composite mass, meaning that it's pretty much a single cell with the tons of the nuclei in the cytoplasm, and you can clearly see it. So yeah, that's quite interesting. So here we're looking at the fixed part of the amoeba. That was actually a live amoeba. I had a picture taking. This is the nucleus. You can see the pseudopods, of course, some of the vacuoles, but in this particular case, you can clearly see the cell membrane, the cytoplasm inside, and this is what you probably will see on the lab practical, one of those particular pictures. Here we're looking at the red as well as kind of grayish colored amoebas. Why this one is red, why this one is grayish, this one is kind of yellowish. Different pigments, it has been specifically stained for you. So here we're looking at the Plasmodium mold. It will feeding Plasmodium, and here's the picture of the feeding Plasmodium. Creating, once again, coordinating, then they create this sporangium, they create the spores, and so on. So, and this is the picture, yeah, this is the picture of the creature, which that's, of course, is the need to change that, but behind my house in a warm and the shady area, it's periodically sprung up whenever there is like rain or anything like that. It will feed off that particular mulch, and then it will create the spores. Spores will go right here, and then it will wait until the next rain or so. Now, next group or last group, we talk about that Apistoconta. We'll look at this particular cell. This particular cell shows the clear cell membrane, nucleus, flagellum in a very curious color. That particular color, this particular cell lives by itself, and then some of them are colonial, and that particular group most likely give us the branch to the animals. As a matter of fact, later on in the unit 3, when we'll start talking about the sponges have something which is known as the colored cells. Inside of the sponge, there will be a structure very, very similar to that. So that's one of the reasons why we think that that particular group, and of course, DNA-wise, we are pretty close to all of the Apistoconta group, including the fungi, but specifically Flagellates. And we are done today with the lecture. So I was able to catch up.